Hello and welcome to another Botany 199 video. In this edition, we'll briefly review the purpose of our antioxidant experiments, our experimental procedures, and our results. Then we'll analyze our datasets, visualize our data, and draw conclusions about the antioxidant capacities of our plant extracts. So let's begin. Free radicals are oxidants. They can remove electrons from compounds like proteins, lipids, and DNA in a cell. Some examples of these reactive oxygen species are shown in the upper graphic. These molecules are byproducts or intermediaries in the process of cellular respiration and build up over time in virtually every cell in our bodies. They are also formed by exposure to environmental mutagens such as UV light or cigarette smoke. Some free radicals have been shown to have roles in regulation of the cell cycle, while the buildup of excess reactive oxygen species in cells over time has been associated with many chronic diseases of aging. The cartoon below depicts this process called oxidative stress. Antioxidants are molecules that can neutralize or scavenge free radicals in cells. They work by donating electrons to free radicals and thereby inactivating them. In the upper cartoon, you can see the red free radical molecule about to damage a stable molecule of, let's say, DNA, shown in green. The antioxidant molecule is shown in yellow. It is able to donate an electron to the free radical and neutralize it before the free radical damages the DNA. Cells produce some enzymes that work as antioxidants to neutralize free radicals. Some vitamins also perform similar functions. Many antioxidants are obtained through the diet, especially plants. Compounds from plants, called phytochemicals, exhibit incredible antioxidant capacities in the lab. Of particular interest are the polyphenols, compounds that play various biological roles in plants. Some examples of polyphenols are shown in the diagram below. The potential health benefits of antioxidants in the diet has led to increased research into the subject. The market for such products is huge and growing rapidly. The purpose of our experiments with Dr. Lee was to assay the antioxidant capacities of local plants to see if they have any potential as dietary additions or sources of supplements. Here is a list of the plants we selected for examination. Many of them, if you remember, came from our garden. And here is a list of procedures, which can be found in our Google Classrooms. It was important for all of us to follow the same procedures with our plants so that our results would be directly comparable to each other. After some background research, we selected our experimental plants and decided which plant parts to examine. Here at 12 o'clock, we see some uki berries. We put our cleaned materials into a food dehydrator and dried them at low heat for several days, at about 130 degrees Fahrenheit. The low heat was to ensure that we did not denature the antioxidants in our plant cells. After drying, we grinded the material in a coffee grinder and weighed out 1 gram of material. We added the ground material to a centrifuge tube and extracted the antioxidants using 10 milliliters of methanol, which is shown at 6 o'clock. The slurries were spun at low speed in a tube rotator for 48 hours. Finally, we spun down the tubes in a centrifuge at 4,000 RPM for 10 minutes to separate the remaining solid materials from the solution. We removed the supernatant for testing and discarded the pellets. All our antioxidant assays were colorimetric. The results could be determined by changes of color in a solution. We used a spectrophotometer to make accurate measurements of these color changes. In the first figure, we see that a spectrophotometer is a light-proof box with an internal lighting and sensor system. A strong white light is shined through a movable prism, which allows the user to select various wavelengths of light for study. The monochromatic light is then directed through the sample, which is held in a clear glass container called a cuvette. The light that passes through the sample is collected by a sensor and can be analyzed. In the bottom figure, we see an example of a spectrophotometer from Dr. Lee's lab. One of the assays we performed was the DPPH radical scavenging assay. DPPH stands for 2,2-diphenol-1-picryl hydroxyl. DPPH is a compound that is composed of stable free radicals, and you can see the chemical structure to the left here. The graph below shows that DPPH is dark purple in color when active, but changes to a yellowish color when the free radicals are scavenged by antioxidants. Notice the peak of color at about 517 nanometers when the free radicals are neutralized. To perform this assay, we prepared two types of tubes. First were our control tubes that contain DPPH and methanol. Methanol, if you remember, was a solvent in both our extracts and the DPPH solutions. The second type of tube, the experimental tubes, contained DPPH and our plant extracts. We inserted these tubes into the spectrophotometer and measured absorbance at 517 nanometers to determine the percentage of DPPH molecules that had been neutralized. We ran the experimental tubes in triplicate and used the control tube as a baseline for comparison. Once again, a document containing all these procedures can be found in our Google Classrooms. So how do we analyze our data? We express the results as the percentage of free radicals that have been neutralized using a simple formula shown here. The percent inhibition of the DPPH radical is equal to the absorbance of the control minus the absorbance of the sample divided by the absorbance of the control times 100. Okay, so here's a real example of how to analyze your data. At the top of this image, you see the raw data. A0 is the absorbance at 517 nanometers for the methanol control, while AS is the absorbance at 517 nanometers for the samples. And as you remember, we ran the experiments in triplicate. So first, you calculate the percent inhibition of DPPH for each trial, as shown in the middle. Then, as shown on the bottom, use Excel to calculate the average or mean, as well as the standard deviations of the measurements. There is an Excel template in our Google Classroom if you need a guide. Finally, use your Excel data to create a simple bar graph showing the mean percent inhibition of DPPH for your extracts plus or minus the standard deviation. Make sure to label your graph properly as shown in this example. In this case, we can see that the extract from water leaves was able to neutralize about 60% of the DPPH free radicals in solution. As a way of comparison, a brief Google search will reveal that a far less concentrated extract of blueberries is able to neutralize over 95% of DPPH free radicals. Our second assay was used to determine the total phenolic content of our plant extracts. Recall that polyphenols are the largest group of antioxidants in plant cells, and that they are important to plants in many ways. The Folin CO-Col2 reagent, called FC, contains compounds that change a solution to a blue color in the presence of these phenolic compounds. To perform this assay, we added 200 microliters of our plant extract to the FC reagent solution and used a spectrophotometer to test for the presence of a blue color at 765 nanometers. In the image below, you can see some of the lab equipment used to carry out these experiments. So how do we use this to analyze our own data? Here's an example using a dataset that was shared to our Google Drive. In this case, we analyzed the total phenolic content of mamaki leaves. The absorbance values at 765 nanometers are shown at the top of the image. These are the raw data. The mean absorbance is 1.237. Well, how does this compare to the standard gallic acid? To estimate this, we use the gallic acid activity standard curve that we produced earlier. Look for an absorbance value of 1.237 on the y-axis. Which concentration of gallic acid is this equivalent to? It looks like it's about 375 milligrams per liter of gallic acid. We can therefore express our value as 375 milligrams of gallic acid equivalents per gram. As a means of comparison, some raspberry varieties have been shown to have values higher than 2,600 milligrams of gallic acid activity equivalents per gram. So what's next? Make sure you've uploaded your group data to our shared Google Drive. This is worth 5 points. Next, create an Excel spreadsheet and associated graphs to analyze your data and upload it to our Google Classroom. An Excel template can be found in our classroom if you need help with this, and this is worth 10 points. Finally, write a half-page conclusion that summarizes your findings. And you can work in a group on all of this stuff, or you can work by yourself. Make sure that all of your different documents are properly labeled if you want credit. Now, I've asked Dr. Lee to send us a comparable data set from other plants. This will give us an idea of the overall antioxidant potential of our plants relative to other more well-known examples. And this is going to be with 5 points, okay? So we're also going to have an online quiz on this material to finish up our project for this year. This dietary nutrition stuff can seem complicated, especially when measuring activity at the molecular level. But as Michael Pollan, author of The Botany of Desire, sums it up, eat food. Real food, that is, not the processed stuff. Not too much, mostly plants. Thanks for listening. Stay healthy, and I'll talk to you soon.